Welcome students to biology class. I shall be dealing with your first class in botany. Few other classes will be taken by your zoology teacher. After that, actually we shall be studying about the plant classification and specifically the 5 kingdom classification. Your zoology teacher must have told you about what is classification, what is taxonomy, what are different types of classification and what is need of classification. However, for your convenience, for your memory recall, I shall be telling you about the taxonomy. What is taxonomy? Taxonomy is the science of naming, describing and classifying organisms and includes all plants, animals, microorganisms of the world. And classification is the arrangement of organisms into groups, categories for a clear understanding, proper study and effective organization. So, you know students, there are millions of plants and animals as well as the microorganisms, the organisms which we see or do not see, they exist in the earth, and it is suggested that 10 to 12 million plants and animals or living organisms are there in the earth. But so far we are able to study in detail about only 2 million, maximum up to 2 million, some people say it is 1.7 millions. We are able to describe in detail these plants, animals, microorganisms, assign a name. So, we give a name to the plant or animal or the microorganism that comprises of a genus name and a species name. Then assign it to different plant groups. The plant groups, or we say as the taxon, plural we say as taxa, are of different types. We start with the smallest group, species, and then comes the genus, family, order, class, then kingdom. Like that we proceed. So, we understand very well that there is a branch of botany or branch of zoology, we shall say branch of biology in general, we can say that deals with naming of the plants. So, we give a name. There is specific rules and regulations that governs naming of plants, animals and microorganisms, and then find out the characteristics. When we classify, we see what are similarities among the living organisms we study. So, we consider similarities, we try to find out what are the differences in their morphology, in their internal structure and in their reproduction pattern, all that. And accordingly, we classify these millions of plants into different groups. Since then, when we have started studying about biology, the great philosopher, scientist Aristotle for the first time, have given a classification of the living organisms, and he has classified the living organisms to two groups, the plants and the animals. And after that, actually we have been classifying in different ways, taking different aspects into consideration. So, we after this first classification of living organisms to plants and animals, Carolus Linnaeus, the genius in biological sciences, has given a 2 kingdom classification. He has put all the plants in the name of plenty, and all the animals in the name of animalia under two kingdoms, plenty and animalia. Then comes the three kingdom classification, taking into consideration the morphological complexities, the internal structure, the tissue system, division of labor and mode of nutrition, and that gives the three kingdom classification. And after that, Copeland proposed the four kingdom classification in the year 1956, which included Monera, Protista, Plantae and Animalia. After that, actually developed the first five kingdom system of classification, which was proposed by Robert Harding Whittaker, or R. H. Whittaker we say, and that was one of the most followed classification. And in this classification, he has, in this five kingdom classification, he has classified the entire living world to five kingdoms, and these five kingdoms are based on certain characters like structure of cell, the plant or animal we are considering is a prokaryote, is having prokaryotic cell, eukaryotic cell, having a cell wall or without a cell wall, and then how do they have nutrition, the pattern of nutrition, whether these are autotrophic or heterotrophic, whether they can synthesize their own food or depend on already synthesized organic matter. Then consider the interrelationship between the organisms and body organization as well as the reproduction. Taking these few important aspects, body organization and reproduction. You know, students, there are different methods of reproduction, like we have sexual, asexual as well as vegetative method of reproduction. Taking into consideration all these aspects, he has given a classification. R. H. Whittaker has given the five kingdom system of classification. So, he has given the five kingdom classification. The first kingdom is referred to as Monera, the second kingdom is referred to as Protista, then the third kingdom refers to fungi, the fourth one Plantae, and the fifth one is Animalia. So, here he has put all the prokaryotes under this group. So, if we see here, we have taken into consideration the structure of cells. All the members of Monera are prokaryotic. So, how do we define a prokaryotic cell? Prokaryotic cells are primitive cells where we shall see there is no nucleus, true nucleus is not found with nuclear envelope. So, in this case, this is a true nucleus. In normal plants, we shall get true nucleus, but now if we consider these members of Moneran group, we shall see a double stranded DNA molecule, normally circular. So, diagrammatically if we represent both the ends of the DNA join and they form a circular DNA. there is no nuclear membrane. Also, there is absence of membrane bound organelles. There is no membrane bound organelles like mitochondria, chloroplast, etc. So, we shall see here this eukaryotic cell or a true cell having a true nucleus, and this nuclear membrane encloses the genetic material which is DNA, and then the DNA remains in combination with that of the proteins. Particularly, these proteins are referred to as the histone proteins, and the DNA along with histone protein, they make the chromatin material, and when the chromatin material passes through the phase of division of the cell, that becomes chromosome. So, we shall see this monneuron group is including all very simple unicellular prokaryotic cells. Unicellular, they are always single celled. Prokaryotic, their nucleus is not developed, they are primitive nucleus represented just by DNA, and then absence of all the membrane bound organelles. Then we shall go to the second group of classification, that includes the protista, and here we shall see this comprises of all unicellular eukaryotic cells, and I have already explained you what is eukaryotic cell. All the eukaryotic cells do have a true nucleus. The nucleus is enclosed by nuclear envelope, and inside the nucleus remains the nuclear material, which is the chromatin material. Then there are membrane bound organelles like the mitochondria, chloroplast, as well as the endomembrane system existing in them. Chromosomes are organized with DNA as well as the histone proteins. You know, 5 different histone proteins join with that of the DNA form the nucleosomes, which form the chromatin material, and the chromatin material upon much folding are forming the chromosomes. So, cell wall is cellulosic. Here I want to mention one thing, that these members of protista, their structure of the cells, if we consider they are eukaryotic, they do have also cell wall, but these cell walls are made up of murine, or we shall say peptidoglycan, and in eukaryotes it is cellulosic. Cellulose, hemicellulose, as well as the pectic substances. And reproduction, we shall see here we shall have only asexual reproduction. So, mostly the members of moneuron group, they divide and redivide just by a method of reproduction, we say binary fission. Whereas, when we go to the eukaryotic cells, that includes algae and many other plants we shall be studying. There we shall see both sexual as well as asexual, and sometimes also vegetative method of reproduction. So, we shall see these are either autotrophic or heterotrophic. They may synthesize their own food, sometimes they are also depending upon already synthesized organic matter. They make the planktons, that may be phytoplankton, that may be zooplankton. They may have appendages like cilia or flagella, which are the locomotory organisms or the pseudopodia we find in case of amoeba and protozoas. Reproduction, which I have already told you, includes sexual and asexual methods, that includes diatoms, protozoans like amoeba, paramecium, etc. Well, the third kingdom in the 5 kingdom classification proposed by Whittaker is the kingdom fungi. Members of the group are heterotrophic, they may be saprophytic, they may be parasitic, or in general we shall say heterotrophic. They cannot synthesize their own food. Cells are eukaryotic. Cell wall is made up of chitin. It is different from that of the eukaryotic cells. The members of Protista, which are made up of cellulose, hemicellulose or pectic substances, or we shall say cellulosic, and the kingdom Monera, where the cell walls were made up of murines and peptidoglycans. Here the thallus is called mycelium. The mycelium is having hyphae, and fungi also remains in symbiotic association with some algae, and they form lichens. Mushroom, mucor, penicillium, etc. are the examples of fungi. Then we come to the fourth kingdom in the 5 kingdom classification, and we start with the kingdom Plantae, which carries very important role in the ecosystem. So, this includes all photosynthetic eukaryotic multicellular organisms with a cell wall what is made up of cellulose. So far we have studied the Monera, which are unicellular. Monera, members of Monera are unicellular. Members of Protista are unicellular. Then fungi starts from unicellular to multicellular. Some are even cynocytic. When we refer to cynocytic, that is a cell with many numbers of nuclei. These are plants which use already synthesized organic matter. Okay. They cannot synthesize their own food, right. So, this comes under fungi, but when we come to the plants, they are all multicellular. So, multicellular plants, there is division of labor, there is organization of tissue system, as well as the organs, and there are 3 organs that make a complete plant. These are autotrophs, and there is synthesis of food by the process photosynthesis. You know, photosynthesis is a process which uses the pigment systems present in the cells, pigment systems, and trap the energy from sunlight, and then takes the raw materials like carbon dioxide. So, we shall say 6 carbon dioxide, 12 water molecules, releasing carbohydrates, regenerate 6 molecules of water, and then 6 oxygen are also been produced. So, this is the process of photosynthesis. Here we do not find any other organism like the members of Monera, Protista or fungi can do this. Some members of Monera may be autotrophic, as we have discussed. They may have some chlorophyll in them, like the bacteria is having bacteriochlorophyll and bacterioviridin, using which they can synthesize this. And also there are some microbes which are chemosynthetic in nature. Then we have gone through these plants, where we shall see there are different types of plants, and we have classified them to Thallophyta, thallophytes, then gymnosperms, pteridosperms, bryophytes, pteridophytes, gymnosperms, and angiosperms. Thallophytes are plant bodies which are not differentiated to leaf, stem and root-like structures. So, Bryophyta includes small plants, where we shall see the most developed one shows almost differentiated plant body, but they are not actually differentiated. However, there is presence of leaf-like appendages, stem-like axis and root-like rhizoids. Then if we see here, these are not properly differentiated, because this bryophyte, they do not have the vascular tissue system. Vascular tissue system has for the first time has appeared among the pteridophytes. These are also referred to as the vascular cryptogams, where we shall see xylem and phloem for the first time entered into this. Then comes gymnosperms, which are having naked seeds. Seeds are not enclosed in the fruits. So, these are the naked seeds, okay. Naked seeds not enclosed in fruits, and then we shall have the angiosperms, where the seeds are born inside the fruits, and we say these are been further divided to monocotyledons and dicotyledons. If we go to the other characteristics of these, we shall see they may be very small in size, microscopic to macroscopic, and small celled, few cell structure to billion cell structures. Then the last kingdom, or the fifth kingdom in the 5 kingdom classification, is kingdom Animalia, and this kingdom includes organisms which are multicellular, eukaryotic and without cell wall. You see, there is a basic difference in the cellular structure of plants and animals, that the plant cell are having cellulosic, chitin or peptidoglycan made cell walls. Cell wall is a must with any type of plant cell, and animal cell is without cell wall. But if we consider the other aspects, they may be uni-to multicellular, but Animalia of Whitaker includes all multicellular, eukaryotic. The nucleus is advanced type. There is a nuclear membrane which encloses the nuclear material or chromatin material, and there is no cell wall. They show all the membrane bound organelles, they show a heterotrophic mode of nutrition. All the members of the group Animalia, they depend on already synthesized organic matter. They ingest it, digest it in their digestive system, and there is absorption by the cells. There may be simple or very complex body structures with specialized tissue differentiation and body organs. So, the animal kingdom or Animalia is divided into many phyla and classes. The examples are Porifera, Coelenterata, Arthropoda, Echinodermata, Chordata, etc. And for example, you can say hydra, starfish, earthworms, monkeys, birds, etc. They include all the animals we find over in the world. And viruses, viroids, and these are not included in the 5 kingdom system of classification, because they are not living cells, they are not cellular. Okay, as they are acellular, they are not put into any of these 5 kingdoms, because we know the characteristics we take into consideration in the 5 kingdom classification does not fit these. 4 of the 5 kingdom consists of eukaryotic organisms, that is only the Monera comprises of only prokaryotic organisms. Then we shall come to the merits and demerits of the 5 kingdom system of classification. The separation of prokaryotes as a different kingdom is very useful and fits into the contemporary understanding of various organisms. This classification is helpful in understanding the progression and development of cellular organization, mode of nutrition, and this classification remains in par with and support the theory of evolution proposed by Charles Darwin. Certain organisms under Protista are not eukaryotic, rather mesokaryotic, and we shall come to the demerits of the 5 kingdom classification. The classification of kingdoms Monera and Protista are problematic. For one, both the kingdoms include organisms with and without cell wall, and one that are autotrophic and heterotrophic. Viruses do not have a classification, they are not included in this 5 kingdom system of classification. Slime molds, actinomycetes, in spite of being considerably different from the rest of the counterparts in the kingdom, have been classified under the Protista. Well, with this we shall go into the details of the 5 kingdoms. Let us start with the Monera. It is also known as kingdom of prokaryotes and consists of all unicellular prokaryotic organisms, that includes eubacteria, cyanobacteria, and archaebacteria. We see here the eubacteria are the bacteria we see in common, and cyanobacteria are the blue-green algae. And the archaea are the primitive forms of the bacteria, which have some resemblances with the bacteria and some characters are different. When we shall go into the Monera in detail, we shall be considering about that. And all members do not possess membrane bound organelles, that is the characteristics of a prokaryotic cell, absence of true nucleus, and then absence of membrane bound organelles. So, membrane bound organelles are absent. Rigid cell walls, you see these cell walls, we have a very defined cell wall. This is made up of murine or peptidoglycan. In the genetic material, that is DNA, there may be any attachment of there is almost no attachment of any proteins. No proteins are found along with this DNA material. Histones are absolutely absent. Locomotory organelles like flagella is present and is made up of flagellin. Flagella is with flagellin, and that is made up of the protein flagellin. And if we go to the further classes of Monera, we shall see they may be autotrophs or heterotrophs. When we shall say autotrophs, they can synthesize their own food material. Like some bacteria, they may be autotrophs as well as heterotrophs. We shall see the blue-green algae is having pigments and many of them are also parasitic. So, they live on plants, animals and microbes, and they cause diseases to the host while they derive nutrition from them. And most important characteristic of the Monera, they are the best decomposers. So, they play very important role in the biogeochemical cycling when we consider the ecosystem. And the kingdom of Monera is divided into many subkingdoms, and these subkingdoms are eubacteria, the true bacteria we see around, and these are including the next subkingdom Archaebacteria. The Archaebacteria includes some of the bacteria like characteristics which can survive the harsh conditions intolerable to other organisms. Then there are anaerobic phototrophic bacteria. Their characteristics includes photosynthesis that does not use water, as the initial substrate does not produce oxygen as end product. We shall say there are chemosynthetic bacteria also. Cyanobacteria includes the blue-green bacteria or blue-green algae, which carries out photosynthesis with the help of chlorophyll A and B. And then the phylum Schizophyta, which includes heterotrophic eubacteria, that include different types of bacteria like saprobes as well as the parasites. Then we shall come to the eubacteria. The bacteria were first observed by the Dutch microscopist, so who was first discovered a microscope, invented a microscope using lenses. He is Antony van Leeuwenhoek. In 1676, using a single lens microscope of his own design, he was able to magnify these very small organisms, and then he published his observations in a series of letters to the Royal Society of London, and he is considered as the inventor of this bacteria. Then this bacterial cell wall is made up of peptidoglycan. These are having, they are classified to gram positive and gram negative types, and gram positive bacteria have a thick peptidoglycan layer and no outer lipid membrane, while gram negative bacteria have only peptidoglycan. So, it was gram stain. When we stain the bacteria with gram stain, there are two bacteria definitely identified. One is gram positive bacteria and the second is gram negative bacteria. You know, bacterium is singular and bacteria is plural. And here the gram positive bacteria have a thick peptidoglycan wall. So, if we say this is very thick peptidoglycan wall and no outer lipid membrane, whereas gram negative bacteria will have a comparatively thin peptidoglycan layer, but we have a lipid membrane outer to that of the so this thin lipid membrane is present outside this. So, this is the basic difference between these, and broadly we classify all the eubacteria or true bacteria into gram positive ones and gram negative ones. They always occur singly, but they may be found in groups. If we consider an alga, blue green alga, you can see they may be occurring, they are independent bodies. Single cell is a cyanobacterial cell, it can exist independently, it can multiply itself, but they may also remain in a colony. We shall find several of them make a colony. Maybe we have several of these cyanobacteria, they are having a common mucilaginous sheath, but each cell is an independent cell. And then if we go to the next point, then we shall see, you can see this is the common bacterial cell, where we have the cell wall made up of peptidoglycan. Then there is you can look to the diagram and you can see also here. So, here remains the nucleoid, this is not nucleus. In the more or less central region of the bacterial cell, there will be a DNA molecule which is double stranded and circular. A DNA molecule can be linear, but when both the ends are joined, 3' end and 5' ends, they join, they form a circular membrane. This is the peptidoglycan layer. We may have a lipid membrane outside, we may not have, and then there are certain projections that may be flagellum made up of flagellin protein, which helps in locomotion. In addition to that, there are certain projections which are referred to as pileus, plural we say pili. And then we shall see they do not have the membrane bound organelles, and there is presence of cytoplasm. We cannot say exactly cytoplasm, however, because there is no nucleoplasm present here. However, the part of the contents of the cell which are thicker towards the cell wall, but comparatively thinner towards it. In addition to this, we shall have some extra nuclear DNA in very small circular DNA molecules we refer to as the plasmids. And if we look to the distribution of this true bacteria, these are widely distributed in air, water as well as land. These are also found in the body of plants and animals, and this can survive under drastic conditions like -17 to about 80 degrees centigrades in presence or absence of oxygen. There are bacteria which can survive in presence of oxygen, and there are bacteria which can survive in the absence of oxygen also. Those which survive in the absence of oxygen, they are referred to as anaerobic bacteria. Well, then we shall go to the shape of bacteria. Bacteria occur in different shapes, and according to the shape of the bacteria, we classify them to different types. The first type of bacteria we shall consider is Bacillus. Bacillus is rod shaped bacterium. It is rod shaped bacterium, and they may occur single or they may occur in twos, they may occur in fours or in a chain. Accordingly, we name them to be Diplobacillus, Streptobacillus, and then we shall come to the second type, that is the spherical type of bacteria, that is referred to as coccus. These are oval or spherical bacteria which are generally non-motile, and they are the smallest among the bacteria. Cocci occur in different forms. They may occur individually or singly. In that condition we say them to be micrococci. They occurs in a pre-individual state, and then they remain in pairs like the Diplobacillus. We shall see this to be Diplococcus. They may occur in a chain, and these are referred to as the Streptococcus, and they may be also found in the form of tetrads, that is referred to as Tetracoccus, or they may be occurring in irregular patches where we shall say them to be Staphylococcus. They are arranged in a particular cuboidal structure, then we say as Sarcini. So, these small cocci or spherical or oval structured bacteria may occur in 6 different types. Then we shall have the third type of bacteria which are found in spiral or helical structure. These bacteria are helical or corkscrew shaped, or these may be coiled. And there are some other shapes like comma shaped, and this example is the Vibrio cholerae that causes the disease cholera. Stalked rods are also rod shaped with a distinct stalk. The size of the bacteria also varies from very small 0.5 to up to 20 micron long or dia. And if we go to the details of the reproduction of these bacteria, we shall see the bacteria reproduce only asexually. There is no sexual type of reproduction found in these. So, we shall see here the bacteria will grow by multiplying itself by the process referred to as binary fission. in this case we shall see the genetic material, which is just a double stranded circular DNA molecule that replicates into 2 and then one copy of each is going to a site, and then there will be laying down of a wall, and the single bacterium will become 2 bacteria. But when gradually the nutrition is depleting or the bacteria are facing difficult environmental condition, they may form spores. Under that condition, we shall see the cell will develop a very resistant wall surrounding this, then this protoplast will be contrasting, and that will store a lot of food material, and finally, when environmental conditions are favorable, the spores may grow into independent plants. If we say the sexual reproduction, there is no sexual reproduction in bacteria. However, we find genetic recombination or that is also referred to as meromixis in bacteria. Like the sexual reproduction, here recombinants are being found. What I mean to say you, there will be joining of some extra genetic material to the original genetic material of a particular bacterium, which is supposed to be possible with sexual reproduction, is possible not by direct formation of gametes and fusion of gametes, just by 3 different methods. So, genetic recombination or meromixis is possible with 3 different methods, that is transformation, transduction and conjugation. In all these 3 cases, genetic information from one bacterial cell will go to the other bacterial cell, not in toto, but in very small quantities through either plasmid or through mesosomes or through just passing of some genetic information from one cell to other cell. When we refer to transformation, genetic information which may be present in the form of plasmids, like the formation of capsule, then if they remain together with the same culture medium, we shall see they will diffuse out of the cell and enter into the host cell, and then they start to express themselves in the host cell. So, the previous bacterium, which was a capsulated bacterium will now transfer the genes responsible for formation of the capsule to the non-capsulated bacteria. This was given by Griffith. He has shown this in his famous mice experiments. And conjugation, when there are 2 bacteria that remain side by side, have some genetic difference in them, and they have the pileus or pili in them. The pileus joins, extends and joins, forming a tubular structure through which actually this conjugation tube, the genetic material will migrate from the donor cell to the recipient cell. By this actually we shall see this cell will become genetically modified. The original combination will change, like that when we bring about two different plants together by fusion of their gametes, we get different characteristics of both together. And the last one I shall discuss is the transduction. Transduction is carried out through the bacteriophage in their lysogenic cycle. And in the next class we shall discuss more about Monera and other plant kingdoms. Thank you, students. We shall again meet in the next class and discuss more characteristics of bacteria and proceed with other plant kingdoms. Thank you.